every research starts with belief. So research is not about knowledge. Research is really about believing and dealing with what we don't know.
Messenger RNA is a copy of our DNA, which allows the organism to produce the proteins that all living organisms need in order to be able to function. After its discovery at the Pasteur Institute in France back in the 60s, this concept has led to numerous scientific advances and opened up new avenues for scientific and medical research.
The elegance of mRNA is that it is produced by copying the information from the DNA, and after it transfers the information for the production of proteins in the cells, it is destroyed. So it has wonderful features for becoming a drug because it has certain functions and then it's naturally degraded and is not anymore there.
Uğur Şahin and Özlem Türeci are both medical doctors and researchers. They quickly realised the potential of mRNA. Originally from Turkey, it is now in Germany that they are conducting their research on cancer immunotherapy with the hope of using mRNA to treat patients. Katalin Kariko is a biochemist. Born in Hungary, she emigrated to the USA in the 80s to continue her research into how RNA induces immune system activation. At that time, the molecule was of little interest to the scientific community.
Unlikely. People did not believe that it can be a drug. Even those who first described how to generate with the phage RNA polymerase, the messenger RNA in a tube. Even today, they said that at that time and later on, they never thought that it could be a medicine. They thought that it would be a material to experiment in a laboratory, but not like medicine.
mRNA was considered as a non-attractive research molecule. It was unstable. Many colleagues ask me, why are you working with mRNA at all? Yeah? It is not stable, why don't you take DNA? But Özlem and I were believing in mRNA because the key features of mRNA being translated in dendritic cells were so attractive that we thought we have to overcome the limitations and spend many years of research to make this molecule work.
mRNA not only has this part which encodes for the protein information, but it also has non-coding elements, several of them. We tested and identified designs of those elements. Basically what this means, we endowed mRNA with a VIP pass and to basically bypass the defense mechanism of the cell, which would otherwise silence this mRNA.
Uğur Şahin and Özlem Türeci succeeded in combining their methodologies for injecting mRNA with the correct elements for generating the desired immune system response. A long experimental research process which Katalin Kariko also undertook, but on the other side of the Atlantic. Together with Drew Weissman, she succeeded in modifying the mRNA so that it was not destroyed by the organism before having passed on its message.
We combined our innovations in one mRNA molecule which had all innovations. And this mRNA backbone became the vector which was used to develop the COVID-19 vaccine candidates. That means her 20 years of research and our 20 years of research was combined in a single project that gave rise to the vaccine.
We had the confidence that it will work. There were already actually publications out with colleagues also at University of Pennsylvania demonstrating in 2017 that the same lipid nanoparticle formulation, nucleoside-modified RNA against Zika, against influenza, HIV, in animal models, it's working super good.
With the COVID-19 pandemic, messenger RNA vaccine has become a medical reality. Today, these three scientists have not forgotten their original hopes and dreams that led them to where they are now.
Our primary goal when we in our research for many years is to develop personalized cancer vaccines. And the idea of a personalized cancer vaccine is to get the tumor sample of a patient, identify the mutations, and then make an mRNA vaccine for the patient. The challenges for this is to be able to make this really fast, yeah? Because the patient has a cancer, and while we are preparing a vaccine, the cancer is continuing to grow. So we had invested many years into this concept and had established extremely fast procedures. And when we learned that the outbreak in Wuhan will most likely become a global pandemic, we knew that the only thing that can stop, yeah, and save millions of lives would be a vaccine. And we knew that we have a technology, our mRNA technology, actually developed for personalized cancer vaccines, could be used for this purpose.
We were able to fulfill at least a part of our vision, namely to translate our science to something which makes a difference. And the COVID-19 vaccine has made this difference. It also has further motivated and incentivized us to work further towards this vision, in particular in all those other therapeutic areas of dire need. Still cancer, which also is a pandemic and where there is much more work to do, and other neglected infectious diseases. So the vision is still there. It has become broader.
This RNA therapy is really 60 years of incremental improvement and improving the delivery method, improving the RNA, the purity, the structural elements, and that's what we were doing here at BioNTech. Every time I said the important is the formulation. So what now is that if you can formulate and you can incorporate some kind of compounds which will make targeted delivery, means that less side effect, more local expression, more local protein is produced, and systemic injection we could deliver specifically will go to the spleen or go to the liver, go to the lung, or bone marrow and other specific cell types. So that's the future. All of these new targeting opens up a new possibility for therapy. Of course, we have difficulties to deliver to the brain, central nervous system, but we are needing the new generation of scientists who will bring the solution.